Binary fission. Here's again in mitosis. We have binary fission. Prokaryotes. The prokaryotes, bacteria and archaea, reproduce by a type of cell division called binary fission. So in binary fission, the chromosomes replicate, beginning at the origin of replication, and the two daughter chromosomes actively move apart. Here's again for the binary fission. First, you see, identical daughter to the cell depends on the attachment of the duplication of chromosomes to the plasma membrane of the parent. So continue to grow in the cell. Continue to grow in continue to growth of the cell gradually separate the chromosomes. Eventually, the plasma membrane grows inwards to divide the cell in two as new cell wall in deposit between the daughter cells. So that's what it does. First, what happens is, like, first of all, as you see here, this is the origin. Then again, first of all, bacteria or plasma. What happens is like the chromosome duplicate first, as you can see, and also as it duplicates, and then we have cell continuous growth of the cell, and where like division of the cell happens, where it produces two daughter cells. So evolution of mitosis, since the prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. So certain protists exhibit types of cell division that seems intermediate between the binary fission and mitosis. Here again, what we have, first of all, we have, during the binary fission, this is the evolution, this is the hypothesis of evolution of mitosis. We have during the binary fission of the bacteria, the daughter cells, the daughter chromosomes are attached to plasma membrane. Elongation of the cell separates, elongation of the cell, the elongation of the cell separates the chromosomes. And then we have here in all eukaryotes, the nucleus contains multiple chromosomes of the cell. So then in cellular algae called dinoflagellates, chromosome attaches to the nuclear envelope, much as the bacterial chromosome attaches to the plasma membrane. Then microtubules passes through the nucleus inside the cytoplasmic tunnels. Then after that, the microtubules functions simply to reinforce the spatial orientation of the nucleus, which then divides the fission process of fission process of of bacterial reproduction. Here's the dinoflagellates. Here again we have here another group of unicellular algae, diatoms, which is diatoms. So the nuclear envelope also remains intact during cell division, but in these organisms, the microtubules forms a spindle within the nucleus. Microtubule separates the chromosomes, and the nucleus split into two daughter nuclei. This is when this is where the spindles forms, and which is placed to this. So then here again we have in most of the eukaryotes, includes plants and animals, the spindle forms outside the nucleus, and the nuclear envelope breaks down during mitosis. Then microtubule separates the chromosomes, and the nuclear envelope then reforms. That's what happens then mostly in eukaryotes. This everything so far we talked about. Here again the concept of the eukaryote cell cycle is regulated by a molecular system. So a frequency of cell division varies with the type of cell. This cell cycle difference results from the regulation at the molecular level. So the cell cycle control. So we have sequential events of the cell cycle are directed by distinct cell cycle control system, which is similar to clock. The cell cycle control system is regulated by both internal and external control. The clock has a specific checkpoint where the cycle stops until a go-ahead signal is received. For many cells, the G1 checkpoint seems to be the most important. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S, G2, and M phases. If the cell does not receive go-ahead signal, it will exhibit the cycle, switching into the non-dividing state called, the G0 phase. Here again we have, the cell receives, a go-ahead signals, where the G1 a cell does not receive. So if it receives, the cycle continues. A cell does not receive a go-ahead signal, it will just, go to the G0, where the cell, will not, divide anymore. Here's the checkpoints. Here we have the three checkpoints are the M checkpoints, the G2 checkpoints, and G1 checkpoints. So that's what happens. So for example, if G1 checkpoints, what happens, if the cell, grows correctly, so what happens is it allows. So if it does not grow well, so it does not allow, it just go back to G0. So but if it divides, if the cell grows, so then it allows to pass to the S phase. Then S phase then replicates. As it replicates, what happens is it passes to the to the G2, and the G2, what happens after that, what happens to the G2 is that it grows more for cell division, to prepare for cell division. But before that, if it doesn't grow well, it means that it will not go to the M phase. That's what the checkpoint is. That's what checkpoints do. It does not allow, if it does not grow, if the cells didn't grow well in G2 phase, it will not lead to the M phase for cell more division. So the G2 will not accept that cell to pass through. So then we have M checkpoints. Then the cell division, if it does not divide well, it will not let that cell to go through the M checkpoint unless it divides well. The cell cycle clock, the cyclin and cyclin dependent kinase, two types of regulatory proteins are involved in the cell cycle control, cyclin and cyclin dependent kinase. So the activity of cyclin and cyclin dependent kinase fluctuates during the cell cycle. Then we have MPF, meaning that maturation promoting factor is a cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into the M phase. So that's all the function of maturation promoting factor, or MPF. Here we have, this graph shows how MPF activity fluctuates the level of cyclin in the cell. And the cell, the cyclin level rises, throughout interphase, which is G1, S phase, and G2 phases, then falls during mitosis or M phase. Then the peak of the MPF activity and cyclin, concentration corresponds. So the CDK itself is present, at a constant level, but it is not shown. So stop and go signs, so internal and external signals at the checkpoint. An example of internal signals is that kinetochores not attached to the spindle microtubule send a molecular signal that delays the anaphase. So some external signal are growth factor, protein released by certain cells that stimulates other cells to divide. For example, platelet-derived growth factor. Platelet-derived growth factor stimulates the division of human fibroblast cell in culture. And here's again, we have the stem of culture fibroblast, then basics medium, of PDGF, which is platelet derived growth factor, and which cells proliferate. Then we have a basics, we have a basics, medium minus PDF cell fails to divide. First we have the cutup of a sample connective tissue. Here's the the cutup of a sample connective tissue into small pieces. Then we have which obtains, then this is the cutup, then this is scalpels. Then we have which and then obtains suspension of a free cell by using enzyme to digest extracellular matrix. Then we are going to have here without a PDGF cell fails to divide. With the PDGF cell proliferates, or cell continues to divide. Then after that we have culture fibroblast. Whereas of culture, then we can have a culture fibroblast. That's how final works, which is here. This is a fibroblast. Another example of external signals is density dependent inhibition, in which is crowded cells stop dividing. So most animal cells also exhibit anchorage dependence, in which they must be attached to substratum in order to divide. Here's a picture of density dependent inhibition of cell division. Then when we have when we have the first one, if we go here we have when we grow cell in culture, normal mammalian cell will multiply until they form a single layer. So the availability of nutrients, growth factors, and substratum for attachment limits the density of the cell population. If some cells are removed, those are those at the border of the resulting gap will divide until the gap is filled with a single layer. This is the first one. Here we have the anchorage dependence, density inhibition, then where it divides. Then so this will replace until until density population. So until the gaps is filled up single layer. This is what we're talking about here. And we have a B, the cancer cell. This is a cancer cell. In contrast to the cancer cell, cells are tolerant of crowding and usually continues to divide well beyond the signal layer, forming a clump of overlapping cells. So individual cells are shown disproportionately here, larger in this figure. Cancer cells exhibit neither density dependent inhibition nor anchorage dependence. Loss of the cell cycle controls in cancer cell. Cancer cells do not respond normally to the body control. Cancer cells may not need to growth factor to growth and divide. They may, make their own growth factor. They may convey growth factor signal without presence of the growth factor. They may have an abnormal cell cycle control system. I think this is where we stop, and we can come back to it. Thank you.